Development. And so it's my hope that after this talk, you'll be able to see the potential of monoform drug development and maybe some areas within your own companies where you might find some uses for it. All right. So the modeling is performed through a science called Pharmacometrics, which is the quantitative pharmacostatistical analysis to answer drug development, regulatory questions, and influence decisions. Or put another way, it's a way to quantitate drug, disease, and trial information to aid efficient drug development and address regulatory decisions. When developing your trial, you want to approach it in a manner that I would say similar to providing a patient with a diagnosis. You want to look at all the information that you have with the highest level of scrutiny, and you want to make sure that you're taking the best path forward. So, former Secretary of Defense Donald Rumsfeld once spoke about a situation and which can be used also to accurately describe drug development process. And he said that there are known knowns. These are things that we know. There are known unknowns. That is to say, there are things that we know we don't know. But there are also unknown unknowns. And these are things that we know we don't know. Yeah, I got through it. Now, in an effort to answer some of these unknowns, Pharmacometrics can look across the data from all of your studies. And this approach, combined with traditional statistical methodology, can leverage all of the collected data that you have. It's really a tool to illuminate a broader clinical understanding and gain confidence of the probability of success of your development plan. So pharmacometrics and statistics work together as part of a learn and confirm paradigm. With pharmacometrics, we can make more assumptions in a hypothesis-generating exploratory fashion, which then guides your confirmatory trials to where the assumptions are going to be kept to a minimum.
So today I want to talk to you about a case study with some work that we did with Protagonist Therapeutics, which is a small pharmaceutical company in California. They're developing an oral peptide agonist for ulcerative colitis, and they came to us to talk about planning their phase 2 study. And then they did something which I think was very useful to them and was valuable to them, which is they started asking, can we look at all our data quantitatively? Can we leverage this to really understand what we're doing going into phase 2? Can we go into this as well equipped as we can to make the right choices? The approach that we took is we said, of course, let's sit down, let's look at all your data. We're going to first see what you have. We looked at what they had, and we determined that we could look at the blood, receptor occupancy, and receptor expression data, and with that probably create some kind of quantitative analysis across all their data. So today I'm going to talk a little bit about the receptor occupancy part of this story just because of time, but the receptor expression story validates it as well. So the first thing that we did is we modeled their healthy mouse data. So for those of you who don't always look at modeling results, this is a representation of the model by running simulations that take into account all the variability, all the uncertainty, and then overlaid on this are the actually observed data. Those are the circles. So the dotted lines represent the model. So what this does for us is it allows us to have this validation that we are doing a reasonably adequate job predicting the observed data, gives us some confidence in the model as being useful. And that's really what we want the models to do. We want to use them as useful tools to guide us. Then the next thing that we did is we took that mouse model and we translated it to the human. And that's what the red line is with the pink confidence interval around it. confidence prediction area. And then we overlay the actual human data. So this provides us with a validation that our mouse model, when translated to the human, is giving us adequate predictions of what we would see. Then the second thing that we did is we modeled the disease mouse data, but we did this in a way that mathematically linked the healthy mouse data with the disease mouse data via what I'm calling colitis multipliers. And again, here you can see a representation of the model with the dashed lines and the overlay of the observed data on that. So we felt this model was also doing a nice job. Then the third thing we did is we modeled their healthy volunteer, first in human data. And again, a representation shown here of the receptor occupancy model for that, showing that this model, again, was doing a nice job capturing what the data was showing. So then what we wanted to do was leverage all of this together. And here you're seeing the results of the mouse models, the healthy and the disease mouse models. This shows their mean response from the modeling. And then use the mathematical links in these models to apply to the healthy volunteer data, and we were able to then give a prediction of what we expect to see in the colitis patient. So as you can see that the response in the colitis patients is fairly different from the healthy volunteer. And then Protagonist was then able to go back and look at in vitro studies that support that there was a different activated state of the integrin that you see in ulcerative colitis. So now we understand physiologically where this is coming from now as well.
So Cecilia, I don't know if this works, to reiterate a point that you just mentioned, that if you would just use the healthy volunteer data, then your dose range would have been too high?
Yes, that's right, Chuck. And certainly going from phase 2, going into phase 2, or even going into phase 3, finding the right dose is something that we all have actively spent time thinking about if you've worked in drug development. And the idea here is not that this would replace anything, but really kind of help give you some quantitative assistance to glean the most from your data. As you can see in this 2014 paper by Hay and others, there is still a 40% failure rate in phase 3. So that to me indicates that we have a responsibility to try and really improve as much as we can. We have a lot of data. We can try and glean from it as much as we can to help guide us. As you know, we're always trying to pick the dose range by balancing efficacy and safety. If a study dose is too high, it's probably going to fail for toxicity. And if it's too low, it's going to fail for lack of efficacy. Then there are many other things that come into play as we try to identify what is an acceptable dose range. What happened with Protagonist is that, like most indications, the efficacy profile and the compound trying to address it was guided by the inherent physiology. But what they found was that in the colitis patient population, the inherent underlying physiology had changed, and it changed the lower bound there of that therapeutic dose range. In addition, we don't know, but the upper bound may have lowered. So really, what happens is if what was the most efficacious dose is now lower, and the minimum efficacious dose is lower, and the dose at which the effect was maxed out is lower. And we also don't know if the toxicity limiting dose may be lower. So if they had proceeded without doing any of this quantitative analysis of their data, modeling, and trying to look really at all their data together in a way that was not just looking at it trying to put a gestalt in their mind, but actually running some models to try to understand it, they may have probably had too high of a dose range and done a lot of formulations at the wrong dose, too high of a dose, and may have even discontinued for the wrong reasons.
So Cecilia, it sounds like they had some confusing data, there were some unknowns, but are other areas that can be helped using this approach?
Yes, Chuck, there is. I would argue that model-informed drug development really is coming as a very helpful partner with all of clinical drug development. Certainly, as we saw for dose selection, it helps a great deal, whether you're going across into doses that haven't been yet studied. So sometimes you have to interpolate or extrapolate between doses. There's approved drugs at doses that have not been actually studied because they've been interpolated. Sometimes you want to change indication and a study population and maybe link between the two. This is a place where modeling can really help guide what you're doing. Sometimes there's safety or ethical considerations for specific populations, such as extensive or poor metabolizers. And an area that is really a good area for drug development with using models is the pediatric population. So, thank you. Those are some
so
ideas.
that's our brief talk.